Hey guys, so I told you that I would come on and I would do some genetics problems for you and try to help you out through some of your genetic stuff. And as I sat down, I realized that I had no sex-linked genetics problems. And so I threw two genetics problems in there. Well, technically it's one, but there's two components to it. And so what we're going to do today is we'll go through Mendelian genetics problems. We'll go through incomplete dominance, and we'll go through epistasis. Epistasis isn't technically on the AP exam, but it does allow us to see some of the stuff that they could apply to us. So they could explain to you what epistasis is and then ask you to apply that information to a problem. So I feel that it's better to be over-prepared than under-prepared. And then I also have a sex-linked example for you. And then also we're going to do a pedigree. Or I'll show you some pedigree stuff. I wasn't able to find the slides I wanted to use, so it's going to be a little choppy, but it is what it is, and I at least sort of made it by my 8:30 deadline to get y'all going. So I'm going to flip the screen so that you guys can see the PowerPoint that I put together, so it'll be a little easier than me trying to handwrite all these genetics problems. It's just going to be clicks of a button. And I've already uploaded the images for this onto apbiopenguins.weebly.com, and I put all of the videos on there, and that's what I'm going to do is I'm going to keep putting all the videos up there. I'll try to put all of my materials up there so you guys have the resources, okay? So I'm going to flip my camera so we can do these genetics problems together. Ooh, that is real weird. Okay, so the first problem we had, we were doing what's the probability of each of the following pairs of parents to produce indicated. And so what we have are these parents here. We've got a homozygous dominant, heterozygous for those traits, and then heterozygous, and heterozygous. You would call this one a trihybrid because of the fact that it's a hybrid for three different characteristics. And we wanted to know what's the probability of getting that offspring from it. And so the first step for all of these are just going to be to do Punnett squares. Now, I'm a strong believer in doing the small Punnett squares, so doing technically just looking at one trait at a time and then multiplying. Because once you get into more and more traits, it involves you making bigger and bigger Punnett squares. So looking at two traits isn't too bad because it's just a 4x4. But if you go up into you're now looking at three traits, that's an 8x8. Or if you do four traits, it's a 16x16 Punnett square. And that takes way too long to fill in. It's a lot easier just to do a single Punnett square, do the cross, determine what the ratio is, and multiply those ratios. So we're first going to cross the capital A with the capital A lowercase a. So homozygous dominant with heterozygous. So we're going to go ahead and put that on the Punnett square. And again, I'm just going to be clicking through this and explaining it. So all of these will be given to you on that website, apbiopenguins.weebly.com. All of these slides will be up there so you can look at them if you need to reference for anything. So when we fill in our Punnett square, we're going to end up getting half of them being homozygous dominant and half of them being heterozygous, okay? So the probability of getting two capital A's, because remember we were looking for capital A, or homozygous dominant on the A allele. So we'll see that half of the time we'll get that. So we'll have to remember that number for later. So now we move into the heterozygous cross. So if we cross the two heterozygotes, you end up finding that we have a probability of 1/4 of them being homozygous recessive. Okay, remember we were looking for the homozygous recessive on the B allele. Okay. And remember, if you have questions, you're more than welcome to pop them down into that little question box, and I'll check it after each question. And the last part, we now need to look at the heterozygous for the C allele. Okay, so if we do that cross, we fill it in, it end up getting, one homozygous dominant, two heterozygotes, and then, of course, one homozygous recessive. And again, we're trying to figure out what the probability of a homozygous dominant is. So probability of homozygous dominant gives you a 1/4 chance. So the last thing I have to do is I now need to multiply these together, okay? So I need to multiply the probability of homozygous dominant with the probability of homozygous recessive with the probability of homozygous dominant in order to get that ratio out. So we're going to multiply those together. So 1/2 times 1/4 times 1/4 gives you 1/32. So real quick, is there any questions on that problem before I move into our second problem? So again, I'm going to do, that was our complete dominance example. We'll move into an incomplete dominance in just a moment, so you guys can kind of see what incomplete dominance looks like. So going once, going twice. Okay, moving into our second problem. So this problem has to do with that we have incomplete dominance, in which we're going to look at a red allele, or a red organism crossed with a white organism is going to end up giving us a pink organism, okay? And so with incomplete dominance, your heterozygous organism is going to be kind of a blend of the two. And so if your teacher has explained to you that you have a red allele, so if there's the red allele, it's going to make the red color, and the white allele will make the white color. So it is usually dealing with some type of protein or some type of enzyme being made, which we know enzymes are proteins. And so because of the fact that you make both red and you make white at the same time, and neither one is completely dominant over the other, you see a blend of the two, which is why when I mix red allele and the white allele together in a heterozygote, then I'm going to get a pink flower out. So as we said a second ago, red and white gives us pink. And then the second thing of the question that I found was a complete dominance. So we're looking at axial and terminal, okay? So we want to know the phenotypic ratio of an F2 generation resulting from the axial red and terminal white, okay? Now, what I'm giving you of axial red and terminal white is your parent generation. So we first need to figure out, well, what is our F1? So if I cross the red with the white, I'm going to get 100% of them to be pink. See, we have one red allele, one white allele, so this one's going to be pink, 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 pink. So 100% of them will be pink. And the same thing goes over here. When I cross my homozygous dominant for axial with my homozygous recessive for terminal, I'm going to get every single one of them to be axial. Now, they'll be heterozygote for axial, but they will still be axial, okay? So the question was asking about the F2 generation. So I have to cross that organism again. So you're going to allow it to go through a self-breeding experiment. It's going to self-fertilize. And so we're going to allow for that heterozygote to cross with another heterozygote. Okay. So what we see here is that I have my pink flower, of course, crossing. That's going to end up giving me one red, two pinks, and one white. So I hope that that makes sense. So we get one red because we have two red alleles. We have two pinks because I have two that have a red and a white allele together, which makes it pink. And then one that has the two white alleles, which makes it white. Okay. So I should find a 1:2:1 ratio of red to pink to white. So 1/4 of the chance will be red, 1/2 will be pink, and then 1/4 will be white. So we do the same thing with our axial. So again, we're going to cross the heterozygote, which we are pretty much awesome on this. So 3/4 of them will be axial and 1/4 will be terminal. So we move into calculating each of our phenotypes. So I went ahead and just wrote them all out. The 6 different combinations you can make here. You can have a red axial, a pink axial, a white axial, a red terminal, a pink terminal, and a white terminal. Basically, you make every single combination. So when we looked at our first Punnett Square, we could have red, we could have pink, we could have white. So you're going to have to put each of those phenotypes with all the possibilities from your other Punnett Square. And that was axial and terminal. So 3 times 2 tells you 6. So you know there's 6 possibilities of different phenotypes you can make, okay? So as I said before, we're going to now calculate. So I need to figure out the probability of each of these. So the probability of red times probability. Okay, the probability of red times probability of axial tells me the probability that I have that are red and axial. And the same thing applies for the pink axial, the white axial, the red terminal, the pink terminal, and the white terminal. Okay, so we're going to then sub in those the probabilities. Okay, so the probability of red we saw was 1/4. The probability of axial was 3/4. And so you would just multiply that together to get 3/16. And the same thing applies for all of them. So pink we found was 1/2 times the axial, which is 3/4, giving you a 6/16. The probability of white was 1/4. Probability of axial was 3/4, which gives you 3/16. Probability of red was 1/4. Probability of terminal was 1/4, so 1/16. Probability of pink was 1/2. Probability of terminal was 1/4, giving you 2/16. And then, of course, probability of white was 1/4. Probability of terminal was 1/4, which gives you 1/16. So overall, you see we have a 3 to 6 to 3 to 1 to 2 to 1 ratio. Traditionally, you would put that in order, so it would be a 6 to 3 to 3 to 2 to 1 to 1. But again, if it's asking you for a phenotypic ratio, you have to definitely give them what it is. So you would say there are 3 red axial, 6 pink axial, 3 white axial, 1 red terminal, 2 pink terminal, 1 white terminal. Whew, that is a lot to say. Now, again, if you have questions, pop them into that little question box so I can try to answer them as we go. Nobody's asked at this point, so we're going to keep on rolling. So the last question we have is a epistasis question. Now, they took epistasis technically off the exam, but there is still a chance that we could still have epistasis. They can describe to you what epistasis is, and then they can actually give you an epistasis question. So this tells us that our black color is dominant to white, and that dominant is going to be, if you get the dominant allele, then it's going to cause this little yellow band, and it makes the fur on black only this thing called a goatee, okay? And so you'll only see that yellow band if you're in a black organism, which is where that epistasis comes into play, okay? So we're going to do our crosses, okay? So again, we have, they want us to know if they're heterozygous at both loci, what happens. So if we cross it, nothing new here. We're just going to have, 3/4 that will be black, and then 1/4 that's white. And then we're going to find that 3/4 of them will be having that yellow band, and then 1/4 will not have the yellow band. So if we put those together, the same thing we did before, where we have black with a yellow band, white with a yellow band, black with no yellow, and then white with no yellow. And we, again, do those ratios. The same thing we did in the last problem. I'm showing you the exact same work. It's just that I wanted to make sure that you guys can see all the problems. And for those of you who just joined in a second ago, all of this is already worked out for you, and it's already posted for you on apbiopenguins.weebly.com. So easy peasy for you. So again, we'll do the probability of black, which we found was 3/4, and the probability of yellow we said was 3/4. So if you multiply that together, you get 9/16. Same thing with probability of white and probability of the yellow. 1/4 times 3/4 gives you 3/16. Okay. Probability of black was 3/4. Probability with the no white was 1/4, giving you 3/16. And probability of white and the probability of no yellow gives you 1/4 times 1/4, which is 1/16. Okay. So nothing new here. But here's where the problem comes into play. We said that if it was yellow, okay, so if you get the capital A, it produces a yellow band on top of the hair that's only on the black fur. And that's what's going to make that a goatee. So 9/16 of them will be that goatee, okay? But that doesn't occur on the fur that doesn't have black, okay? So we're also going to see that if there's no yellow, that 3/16 are black. But what happens with this one that has yellow on the white fur? Well, the thing is, it doesn't show up on the white fur. So both this one and this one are both going to show up as white. So your final ratio is going to be a 9 to 3 to 4 ratio, because you're going to add these two together because this is your two whites. So you would have 9/16 are goatee, 3/16 are black, and then 4 of them that are white. So again, if you have questions, you want to put them down into that little question box so that I can answer them as we go. I don't really see any questions at this point, so I'm going to keep on rolling because I like to just roll. So I just added this problem in, so it's kind of not as pretty as the other ones are. But it still does kind of work through for you. So we have a red-green color blindness is due to sex-linked traits, okay? For my students, they haven't gotten to sex-linked yet, but I figured if I was making a genetics video, I should have sex-linked materials in it too, so you guys had everything all in one spot. So a colorblind male marries a woman with normal vision whose father was colorblind, okay? So one thing to note is that when we're talking about sex-linked, it's going to be on the X chromosome. Females have 2 X chromosomes, while males only have 1 X chromosome. So knowing that her father has this disorder, knowing that he's colorblind, means that he passed on the recessive allele to his daughter. So we know the daughter's going to be heterozygous because she's normal, but she had a father with colorblindness. So he passed on the recessive allele, and then she has a dominant allele that came from her mother to allow her to be normal. Okay, and it says that she marries a colorblind male. So the person that she marries, that she mates with, is going to be colorblind, so he also is going to have that recessive allele. So if we set this up on our Punnett Square, this is her husband, who is a carrier. So he's colorblindness. So we have a lowercase c here, and of course he's XY because he's a male. And then, of course, we put her over here. And I missed the little underlining here. I try to tell my students to underline ones that look the same, because whenever you're writing them out, if they look the same, the reader's not going to be able to tell whether it's a capital or a lowercase. So put a line underneath your letters that look the same. Like C's look the same, U's look the same, V's look the same, W, Z. There's a lot of letters out there that look the exact same. So you want to make sure that you can denote whether it's a capital or a lowercase. So if we do this cross, which as you see, didn't make it as pretty as I wanted it to, you're going to find that you have one that will be a normal female because of the fact that she'll be a carrier. She's still got the normal allele, and then she'll have that recessive colorblindness allele, but she will look normal. She'll be able to have normal vision. You'll have a normal male because of the fact that he has that dominant allele. You'll have one colorblind female because she has two of the recessive alleles, and then you'll have one colorblind male because of the fact that he'll have the recessive allele. So the question was, what is the probability that they will have a colorblind daughter? So the wording on this is very important. If they say a colorblind daughter, you're looking at the entire Punnett Square, and you're looking at, okay, what's the probability of having a daughter that is also colorblind? So like you're wanting it together, okay? So when we look at the full Punnett Square, okay, the whole thing, there's 4 options. And one out of these 4 options are going to be a colorblind daughter. So we get a 1/4 chance, okay? Now the second question that we have is, what's the probability that the first son will be colorblind? So now I'm saying, okay, what's the probability of having a son and that son being colorblind? So by saying the gender first, by saying that it is a male, a biological male first, we're saying we're only going to look at that part of our Punnett Square, okay? So let me flip it back over. So now we only look at this, okay? So what's the probability that you're going to have a male that is colorblind? Well, that's going to be 1/2 because of the fact that we're looking here, there's only 2 options. We have a normal male and a colorblind male. So 1/2 would be the probability of having a son that is colorblind. Okay? So again, if we have questions, pop them down into that little spot. And so that is actually the end of the little PowerPoint that I made. But I pulled my PowerPoint from my big review from last year, so you guys could see some pedigree stuff, because I asked if y'all needed pedigrees, and you said, yes, do pedigrees, please. So I'm going to just do a little quick thing with the pedigrees, and then we will be on our way. So here, just so we're clear, on a pedigree, if you see a square, the square is a male, a circle is a female, and again, that would be the biological gender for them. So if it's shaded, it means that they're affected. They have whatever the trait it is that we're tracking or we're looking at. If they're not shaded, they do not have that trait, okay? And we're going to see in a second that if we're kind of like half shaded, that means that they're going to be a carrier. So looking at this, I'm able to tell that this is actually a autosomal recessive. Autosomal means that it's on one of the 22 chromosomes that just is the normal body chromosomes that does not determine your biological sex, okay? So it'd be one of the chromosome 1 through 22 is an autosome. And by saying it's recessive, of course, it is not dominant. It's the one that's going to be hidden. You only see it if you get 2 of the recessive alleles, you're homozygous recessive. So we can tell that this is a recessive allele because you have 2 unaffected parents with an affected parent. I'm sorry, an affected child. So 2 unaffected parents are able to get you an affected child. Why? Because that trait was was hidden. It was masked by the fact that the parents were heterozygous. They were able to mask that recessive allele, but they could still pass it on. There's a 1/4 chance of that happening, but it still can happen. And so that allows us to say that, okay, well, this must be a recessive allele. If it was dominant, you will see it in every single generation. Every generation will have it if it's a dominant allele. Okay. Also, looking at autosomal is going to be equally likely for males and females to get it if it's an autosomal trait. If it's a sex-linked trait, we're going to find more males will have the trait. Why? Because males only have one X chromosome. So there's not a second allele that can mask it. So if they get the trait, they have it. If they don't get the trait, they don't have it. So it's pretty much based on that one X chromosome. They get the X, they got it. They don't get the X, they don't got it. Easy peasy. I am pressing the wrong button. So now let's flip it over and let's look over here at the next one. So as I said, it was not really a pretty thing. So as I said before, if it's like a half circle, that's showing you that it's a carrier. So in this one, we can tell that this one is a sex-linked recessive because you kind of need to follow it. Okay, so for example, this is an affected male, and if you notice that there are no affected males from them, they have carrier females, but they have no affected males. And then also you can see here that the carrier female then, of course, passes it on to her son. Okay, because of the fact that in order for a male to have it, they must have gotten it from their mother. So you can see that this mother passed it on to her son. And again, that is showing a sex-linked recessive. And the last one I need to show you is a little kind of funky one. Okay, so real quick, mitochondria comes from your mother. Okay, so when the egg is formed, it's actually this really big cell that has all the organelles needed, and the sperm is very, very small and has pretty much no organelles in it. There has been studies recently that show that there's some DNA that's coming from it, but for the most part, we're seeing that it's the female. And also, straight out of the CED, mitochondria and chloroplast are coming from the female. So definitely know that the mitochondria and the chloroplast comes from the female. Okay, so if we have an affected DNA inside of your mitochondria, yes, you guys all remember that the mitochondria and the chloroplast both have DNA in them. If the affected allele, the affected gene, is in the mitochondria, then you're going to find that the mother will pass it on to all of her offspring. All of her offspring. So if you look here at this Punnett Square, this pedigree, you see you've affected mother, and look, every single one of her children is affected. Again, affected mother, and every single one of her children is affected. Okay, unaffected mother, and look, none of her children are affected. Unaffected mother, and none of her children are affected. So, again, if you see that you have affected mother and all the children are affected, there's a high chance that it's going to be mitochondrial. So, I hope that was helpful. I hope that I'm going to let you have a second. Any questions? I've come up with a new tagline to leave each one of these, so super excited about this. It's not anything new, but those of you who know me know I'm about to say this, so it's okay. But so since there's no questions, I'd like to remind you that you are AP Bio Penguins and you are dressed for success. Bye.